Dear all, my name is Hackman, and I'm going to talk about the challenges involved in determining the pathogenicity of Titin variants. So first of all, I like to say that I'm really happy for being here, for being invited here to Nashville, Tennessee, to the SciFam conference. And also, I'm sorry that I could not attend physically because of some medical reasons. And now I have it like recording online, and this is of course not the same thing. But anyway, so many of you know me from before, so I'm not going into that. But directly, if we jump to Titin, you can say that, okay, it is really a challenge to determine if some variants are pathogenic or not generally, and especially goes for this gene. So you can follow the ACMG and AMP guidelines, but however, because of the sheer size of the coding region of Titin, which is like 100 kilobases, which means 50 to 100 regular genes, rare variants in the Titin genes are detected frequently in any sequencing approach that includes this gene, whatever the clinical hypothesis might be. And also most healthy individuals have at least one unique or rare variant in Titin. That doesn't really necessarily mean anything. So most Titin missense variants identified have unknown significance, so they are WUSIs, and their clinical evaluation in diagnostic laboratories remains challenging. Titin also is a complex, highly repetitive structure of the gene, and that also poses technical problems. So there are variants in, for instance, the triplicate region, and they are often not called or miscalled, just for technical sequencing reasons. And also Titin seems to be relatively resistant to the effect of small missense and other in-frame mutations in most of its parts. So Titin is certainly a special case. So if you think about the pathogenic Titin variants in skeletal muscle disorders, I'm not now going into the cardiomyopathies because they are handled in other lectures. So the dominant TMDM HMERF homomorph associated missense variants in exons are in exons 363, 364, and 344 respectively. Most other pathogenic Titin variants causing skeletal muscular disorders are actually recessive mutations. These are dominant, the one that I mentioned. Then there are these compound heterozygotes with a Titin truncating variants in one and a missense mutation in the other allele causing muscular disorders. Also, the Titin-related phenotype, muscle phenotype, ranges from severe congenital myopathies to relatively mild adult onset muscle disorders. But one thing is in common, and that is that they seem to be fully penetrant. However, some with quite late onset. So if you see frequencies of 2.4 from 10 up to -7 for a variant in a dominant skeletal myopathy, or 9.8 times multiplied with 10 in -5 in recessive diseases, that's a moderate indication of pathogenicity if it's combined with co-segregation of this variant. And combined with supportive in vitro studies, for instance, abnormal Titin fragments and in vivo studies, knock-in models, these are strong evidence. So as you know, this is an old picture of different diseases associated with mutations in Titin. One thing that you can see here that they are concentrated to certain areas. So for instance, the HMERF are concentrated to one exon 344 and TMDs to the two last exons. So there are hotspots, and that also already makes it sort of easier to to predict if a mutation can have an impact or not, because if it's in a hotspot, then it's more likely to have an impact. But there are some challenges if you want to look at the functional things of a Titin missense variant, and that is that the whole cDNA cannot be readily cloned because it's so long. If you want to test for protein-protein interaction, and then you have to use shorter fragments. But actually, this was successful in this TMD in the Finn-Mai and some other mutations that cause the Finnish TMD and the LGMDR10, because we could actually use and see small fragments of Titin that we detected, which was from the last part of Titin, the C-terminal part. So in this case, we didn't have to check the whole Titin. We could sort of monitor shorter fragments. But that was in this case, and it may not be in all cases. But this is easy to remember, good to remember that sometimes you can use, even for Titin and other long genes, you can use this sort of, that you check some shorter fragments of the gene. But however, one thing that is really useful are the Titin databases. And as you may know, in 2015, we began to plan the Titin database. We had a meeting in Leiden about it, but it did not really reach the goals because it was difficult convincing groups to submit enough of their data. So that didn't work out as well as we wanted. But there are these, in Leiden, this database, CleanVaris, of course. Then Gautel has this Titin database where there's like the protein coding regions and then GNOMAD. And actually combined use of available databases is a good solution. And sharing of data between groups is very important. So there's not really any need to now to begin to try to create a new database. And in this case, we should remember that sharing of data is important. So sharing is caring, not scary. Then, of course, RNA-seq is very important because it's now a standard genomic approach to investigate molecular biological processes, and it's very useful for Titin analysis. And the only thing is that again, Titin is so long that when you do the RNA-seq, you should think about if you use the poly-A version versus the ribodepletion version, because with the poly-A, you may not get all of the ending fragments of Titin. Detection in RNA-seq, however, does not prove that the isoform is actually translated, but of course it's good, a good start. Then we have the long PCR, which is a coming thing, and they are now becoming better. Some of the issues that were there are not there anymore, and it's PCR free, so you don't have the PCR related problems. And and you can see from a single individual which variants are on the same allele, so you don't need many individuals to see if a variant is where two variants are on the same or different alleles of Titin. And that's very important for detecting de novo variants, for instance. And there are these different companies that do SPAC-Bio also together with Twist and then Nanopore. And but then you should remember that detection of a compound heterozygosity of a rare variant together with a Trankerian variant can be common finding, and it's only to be considering supporting data. Then we have all of these computational tools to detect copy number variation and structural rearrangements. And they are a good complement to the traditional algorithms aiming at detection of CNVs, SNVs, or small indels. Then we have the VARSOM that I will come to. And then we have the in silico structural studies, this Titin database that can be considering supporting evidence of pathogenicity or non-pathogenicity together with other prediction from bioinformatic programs. And then we have the predictional programs. The predictional programs, there are many of them, but however, they are not actually that, that useful because they are, except especially for Titin, they are not so reliable. Varsom, however, is a variant knowledge community, a data aggregator, and variant data discovery tool, and that is becoming more important. And it's a community-driven project, and it has there are at the moment more than 400,000 users worldwide. So this can be really, really useful. So conclusions is that Titin is a challenging gene due to its sheer size, and most individuals have at least one or rare or unique missense variant. And they pop up in many analyses without any, and may or may not have actually a pathogenic effect, so you should be careful. Most of the pathogenic Titin variants used so far detected have been recessive. Compound heterozygosis with one missense and another truncating has been found, and the current predictive bioinformatics tools are not reliable, as we said. So you need segregation studies, functional tests, protein and RNA-seq to prove that some variant is really potentially interesting. And the use of available databases is important. And Varsom is becoming more important than useful, and long-read sequencing will also become more important. And also knock-in models are still important. Thank you very much. And here's a picture a few years old when we looked younger and more beautiful of our group. And then this foundation that has supported us. Now there's time for questions, please.
The bus